Hi, my name's Andrew Dick, and I'm the pleasure to be the Professor of Ophthalmology in two universities in Britain. One is in Bristol, which is a small city west of London, and the other's at the Institute of Ophthalmology, which is in UCL, London. I'm a clinician scientist, but my real interest lies in immunology, and hence how the immune system keeps the tissues healthy and our tissues functioning. So I was really pleased to be invited to this meeting because whilst I don't see too many patients with macular degeneration, because I'm largely in the lab now or directing lab research, I was very intrigued for over many years of why we didn't engage with understanding the immune system until we had this gene-wide approach that showed that immune system was involved in macular degeneration. So being at this meeting, what I wanted to do is give an idea about how I think the immune system works in macular degeneration. And to do that, I'm thinking that when we look at immune responses in general in our body, it's really to keep us protected from danger, viruses, bacteria, or other insults. And also it's there to keep the tissues healthy. So it's acting all the time so that our cells within every tissue, including the retina, are kept as healthy as possible. So if we now come to macular degeneration, what we see is that the cells in the retina, of which there are many cells that are capable of mounting an immune response, as well as the systemic circulation that drives immune responses, are very active during macular degeneration. But what we don't know is whether that immune response that we see is a protective immune response to keep our cells healthy, to make our cells live longer, i.e. survival of the cells, or whether when the immune response is heightened, it's actually a sign that we're starting to damage the tissue and it's accelerating that damage to the tissue. So it's important to understand the differences because obviously one, any treatment in the future would augment the immune response, or if we thought it was damaging, you'd want to inhibit it. And then we have the complexity of where we are throughout the course of disease. So the idea of what I wanted to get across in the talk that I gave yesterday was that the immune responses are occurring all the time inside a cell, not just outside cells. And when they're occurring inside a cell, it's to regulate the cell metabolism. So the engine room in the cell, the mitochondria. So what we see is when you heighten an immune response within a cell, you're protecting that cell survival by maintaining the health of the mitochondria. And one of the molecules we found that do that is a cytokine or a protein released during immune responses called interleukin 33. And this cytokine basically keeps your mitochondria healthy. So what does that mean? Well, if we're able to detect that in humans early, we could be able to augment that response to keep our mitochondria and the engine of the cell healthy and the cell surviving longer. However, there will become a threshold when we do know those cells are under so much stress that the immune responses we see, whether that's complement mediated or other cytokines that cause damage, what are called cytopathic cytokines, then we'd obviously want to target inhibiting those cytokine pathways. But to do that, we really need to know when that switch is, because there are differences in therapies to enhance immune responses to protect the cell versus inhibiting immune responses to prevent damage is going to be pivotal during the course of AMD. So in the future, where we'd like to go is to understand through imaging technology when the cell is under stress and its metabolism is changing, that we'll be able to enhance the immune responses within that cell to protect that cell and alter its metabolism to maintain its survival. And imaging might be able to help us when we're getting to the stage of understanding when the cells are being damaged, earliest phases of damage, and then understand the pathways where we could try and use immunotherapy to prevent that damage going forward. So it's not going to be a one pathway treats all, and it's very likely to be timing during that macular degeneration going forward. But we are at a time where our technology is going to help us do that. Our imaging technology for biomarkers for the immune response, the ability to understand immune responses through genetic work by doing what's called transcriptomics or epigenomics, to understand how that is altering in cells and also in peripheral circulation will help us understand the timing of future therapies.
How do you see this research translating into patient care?
So two ways. One is both the immune response could act as a biomarker. So what I mean by that is that when you're noticing changes in biomarkers or the immune response system, then that might highlight where we are in disease. So even if we're not using immune therapies, it will tell us when the best time to intervene for progression will be. So I think very soon what we'll be able to do is look at metabolism of cells through imaging technology, and that can come through into the clinic quite quickly in order to understand what the true health, energetic health of that cell is. When it comes to treatments, it's a bit more difficult because I do think that we do need a more stratified approach for macular degeneration, in that whilst we understand it looks like one disease, I think given patient differences and multiple pathways, each patient may end up with a similar phenotype. What I mean by that is how it looks, but by different mechanisms, and therefore understanding one may be more immune mediated than other patients is really important.